take biological data from collections and some functions in it, and we're going to map a little bit these occurrences. I will just go to the, don't share yet, you don't need to share yet. Yeah, oh, it's not good? Peraí, às vezes ele volta. Tá ainda não? Ah, agora foi. Sim, sim, agora foi. Peraí. Ok. Okay, so I think this is here, no, the USB. Sorry, Natalia. Is the volume okay? So I don't have the USB for the pointer. Ah, it's here. Yeah, it's here. Okay. Can go ahead. Okay, so today I'm going to talk about, hi everyone, basic concepts about biodiversity databases for those who haven't seen this kind of data. I'm going to talk about the standard for notation for these databases. It's called the Darwin Core Standard. And we are going to perform some biodiversity data cleaning because these data are super messy when they come from the raw sources. I'll talk a little bit about the errors, the biases, and some steps that we need to do when we clean data. And we are going to use some scripts to keep coding about this. So this is what we're going to do today. I know that some of you are, for example, bioinformaticians in the room. And lots of us in our fields, we have classic data repositories. And in ecology and biology and in taxonomy, our main source of raw data are collections. Collections in the field, collections by taxonomists, collections by people who study the species, who study the classifications, and that have collections with preserved specimens around the world in museums, herbaria, and collections, biological collections. This is a picture of the fish collection at the National Museum in Rio de Janeiro. This is a picture of one of the shelves with the Ecological for plants from the Rio de Janeiro Botanical Garden. But most of us from the universities we come from, there must be similar collections. And in our countries, there are some large, famous, important historic collections, herbaria, and some university-based collections. So everybody has access to this kind of data. And it's biologists in the field who create this data, so researchers, but also there's a lot of training by undergrads and grad school courses who take the data from the field Sometimes the aim of this data is not studying the species by themselves, but for example, you go to a plot, you want to know the vegetation, so you have some collections. Even if your main interest is not the species that are there, you want to see the whole communities, you have some collections, you take them back to the collections. But we have, in the recent years, we have more and more collections that are taken by amateurs, by people who are not necessarily biologists by training. And we have some apps, and we have some, for example, bird watchers have lots of collections of biological data that are not, at the end, preserved, but they are still biological data. Please, can we have a mic here? Here.
So one thing that frustrates me a bit coming from mathematics is that we cannot really have those acts of citizen science. I don't know how you say that in English, but ciência cidadã, citizen science.
Citizen science.
But also from not knowing anything really about how that works, I always have doubts regarding the quality of the data and if it is trustworthy to use in scientific research. How is that in ecology and the likes? Because you mentioned bird watching, and that's the example I mostly think of, because you say, oh, I saw a tucana here, and then you put it there, and how generally people treat those kinds of data? They treat it separately from data collected by organized institutions?
It's treated separately. When you have databases with this kind of data, you can distinguish what is a preserved specimen from an observation. And not every data that comes from apps, for example, goes into the same repositories. You can access those data from another entry point. So at all times you have this distinction about the source of the data. But what you say about data quality is super important, not only coming from apps, because you have some very good bird watchers that have a very good eye, very good experience, so their data are good. And some of the apps have a couple of steps in checking that what you are saying makes sense. But the data, even the data that we biologists take in the field, their quality is very heterogeneous. Sometimes you are in a field course and you don't have a specialist of the family. You have some families that are more difficult than others. You have to have specialists. So every collection around the world has flaws in the quality of the determination, the final name that a specimen has. It could be the world specialist that said this, that put the name, but most of the time it's somebody who's acquainted with the family and not necessarily a specialist. Specialists travel around the world and they check exicatas and pictures, high quality pictures that now are digitalized, and they correct some IDs. So an exicata, one collection, can be updated because somebody said, no, this is wrong and you have to update this. And for those of you who do not work with taxonomy or not acquainted with taxonomy, names also change all the time. So if a specialist says this species is actually this other species, this update has to be reflected somewhere. So when you go to a collection and you see a name, it has a whole story behind. It has quality, but it can also be a synonym from other name, and so we deal with that all the time. So data quality is key here. I think we're going to be back in some places about this because it's really, really messy sometimes. Who uses this kind of data? So not only researchers that are interested in the evolution of groups, and the curators themselves, the people who are in charge of the collections, but also the overall community. And there's a movement to open this kind of data with their pitfalls and with their pros and cons. The ideal is to have standards to open this data. So right now you have portals where you can access data for collections around the world. It's a massive amount of data between what is being created right now and what is being digitalized from historical collections. The repositories are giant, so every collection needs a TI department and people who are in charge of ensuring the data quality, and that takes resources, and it's not exactly the most. When you talk about funding for universities, the collections are not the ones that make the case like, hey, this is super novelty, but more rather a historical thing. So collections do struggle with funding for keeping this data with a good quality. And from this amount of data, there are some large-scale syntheses that can be done. So not only because you know which species is what and where it is. When we study biodiversity around the world, we need this data. We like to know where the species are around the world. So lots of studies worldwide wouldn't be possible if this data weren't there. However, the quality is very heterogeneous, and there's a lot of inequality between countries and regions. Most of the collections that were made in the south of the planet, in South America and in Africa, are right now in collections in Europe. Some countries are trying to repatriate, make some repatriation, so that this knowledge that was outside comes back in a form as every country to know more about their own diversity. Herbaria have a diplomacy system. They change. It's a friendly system. But this kind of information that was historically collected needs to be relocated to their origins. And also, I don't know if you have a duplicata. Every time you make a collection, you can place some duplicatas in a couple of collections. You put one specimen in your local field, the closest collection, one in the most important collection in your country, but also you can send it abroad. So you have, in addition to the errors that you can make, you can have 3 specimens from the same plant in different herbariums being named by different people. So you have to, sometimes when you do it, the data check is like, who did this? So it's a little bit of a prestige. Like depending on who was the person who put the name, you can say, okay, I have some more confidence. And we have lists of specialists. We work with lists of specialists. If a person that is, you can have a database, and if you see the person the person who identified the specimen is a person who is a specialist, you can cross the databases. It's more and more easy to do that. And this is a large field that is called biodiversity informatics. So we manage and lead with primary biodiversity data for many applications. And the worldwide repository of this kind of data is called GBIF, the Global Biodiversity Information Facility. It was created after Rio 92, when the Biological Diversity Convention was held and signed. One of the things that we needed to do as a planet was, hey, what's in there? So the Global Biodiversity Information was created. And some of the countries decided to make local, country-level biodiversity information systems. But GBIF is previous to many of them. And not all countries have their own national biodiversity systems. Some countries go directly to GBIF. They just send the data, the herbariums send the data directly to GBIF, but some highly diverse countries have their own national biodiversity information systems that are linked eventually to GBIF. In Brazil, we have 2. We have Species Link. That is it's organized by a private company. It was before the CBBRE, that is the public one. It's 2 large biodiversity data repositories. It's not like a private, it's like a non-profit, but yeah, it has a different system of funding. In Colombia, we have the CIB Colombia. It's based in the Instituto Humboldt, and they are the ones who are responsible phase 2 GBIF. And Mexico and Argentina, I didn't know that, they also have. But you can come from a country where the collections go directly to GBIF. And there are some countries that participate via their ministries, depending on their organization. So, but the large repository is GBIF. And we have other systems, like the USA has like parallels. It goes, some of the data in the USA goes to GBIF, but they also have like their own networks of data. So depending on what's the region that you want to study, you have your own repositories. And you have iNaturalist, which is one of the repositories of data that comes from citizen science. So you have lots of data sources. You have from flora or by taxonomy group. You have, for example, the fish base, a mammal diversity base that is world. It collects different, for example, mammal collections around the world, and it's a network of specialists. So depending on the group you work, you can have more contact with one or the other. One of the works of the people around the R ecosystem is creating packages that do the interface with these databases. So if you have an API, if you have a database, and you want to access this data via R, like I told you yesterday, there are some packages that have internal data. Some packages can be made to be wrappers to access the APIs, and they create also tools for data cleaning and to make data available and to have some checks. And ROpenSci is one of these large initiatives. ROpenSci has now a community of more than 700 people. They create packages. They have more than 300 or 400 packages. And they do something that is very neat and very useful in biology, that is package peer review. Because most of us are not coders. Most of us are not programmers, but we're doing code. So sometimes you do the code and you don't know if it's working the way it's expected, or you don't know how to code more efficiently. You don't know lots of things. So one thing is to have a function, and another thing is having a function that can be used around the world. And they do this, they do the package peer review. So if you want to put your data or your package in the network, in the internet, in CRAN, they have a network of people who can check if your code is good, if the package is nice, and it's a friendly peer review. It's not like a paper peer review. It's intended to be friendly. And most of the source of biodiversity data in the R ecosystem comes from here. So you will hear about RGBIF, for example. It's the interface to GBIF. Tax size and tax view, we have lots of taxonomical backbones who will deal with the synonyms, with the changes in names. And some packages that deal with the formats, with specific formats. Today we're going to use GBIF and DecoStand, that is, no, TaxonStand, yeah. Yes. Sorry? Okay, you're like, what?
I have a question. It's like this database is, as you said, that the data is very messy and you have to clean everything. And it's more like a curiosity, because sometimes different people clean the same data set over and over again, because you clean it and then you use in your analysis, and that stays there, or at the best you put in a repository and the person has to search for your article to find that data that you cleaned. So how will you deal, because this databases, there's no one like doing some curation or something like this. And many people clean the same thing a lot of times, and it seems like a huge effort that should not be reproduced all this time. You know what I'm saying?
Yes, absolutely.
You have any? Do you know any initiatives or something to deal with this thing?
Yes, I think the people at GBIF, like the original way of working of GBIF is collections go directly to GBIF, and from the collection there's an update. But if you do a revision and you take a data set and you clean it, or you change and you edit it and you leave it clean, in part I am just going to make a parenthesis. The coordinates, for example, in the ECOLOGY sometimes are in different formats. Some of them are with N and S, with the classical notation in degrees. Some of them are in decimal. The newer collections are taken by GPS, so they have like very good coordinates, but coordinates were a mess before. If somebody takes a new data set and cleans it and puts everything in the same pattern, they can publish the data set either via, for example, Dryad, via a repository that goes with a data paper or with a paper, but also GBIF has a way for you to publish the data set. There's another entry point in GBIF. It's called the IPT. I have one slide about the IPT only because we're not going to deal with IPT data, but you can submit your data set to IPT. It will have a DOI, it will have metadata. You can explain the methodology, and they will be happy to host it. The problem later is how you publicize this, because sometimes you reclean data because you didn't know nobody did it. So this is a question of communication. Most people do not know somebody cleaned this. But the culture of receiving clean data sets is already in the IPT of GBIF. It's just another database. You want, if you look for data in GBIF, you won't have this access, but it's another way. But yes, it's important about this. What you say is a very common problem. Data papers are a good option, but still. So we are going to look at GBIF data. I have a tutorial, but I am afraid that if you open right now the tutorial, it's going to be like a loop and you don't go out of the tutorial. But this is basically what we're going to do at the beginning. We're going to download RGBIF, install RGBIF, and make a download of a data set. I picked Myrsina coriacea because it's a widely distributed species in South America. But we could do this with any species. For now, I will go to a tutorial and go with you. But basically, when you have this kind of data, the column names, there's another problem. The column names must be in a standard. If I say this is latitude, and the other person says it's latitude in their own language, you don't have even the column names. They are not even standardized. So when they created GBIF, they also created a standard for this kind of data. And this standard is called, it's worked in the Darwin Core group, and it's called the Darwin Core standard. And every person who leads with biological data should be trying to attend these standards. I think you have access to the links in the page, okay? So I won't click anything, but we can go back if you have any doubts. And possible fields are, of course, the taxonomic information and the metadata about the locality, the geographic coordinates, but also who collected, when did they collect, how did they collect, what was the method, and what species attributes. When we go to a field, we say that the plant was 8 meters high, it was under the shade of a large palm tree. We write lots of things. And there's space in these databases for your own field notes. You can put them in these databases. We are not going to go to the IUCN status, but the International Union for the Conservation of Nature has a whole protocol to assess when a species is going to be extinct or not. Have you ever heard about the IUCN status and the red lists? So, whenever there's a news about some species on the verge of going extinct, there was IUCN evaluation. And this is centered in the UK. The IUCN is in the UK, but also we have the same system. Countries have IUCN authorities. And in Colombia, for example, it's again the Humboldt. Here in Brazil, it's the CNS Flora at the Rio de Janeiro Botanical Garden, where I was. It's the national authority for plants and red lists. So you follow the protocols in your country, and you can put this information on the top. So if a species is locally imperiled or if it's globally imperiled, it's like the whole repository for extinction risk. And this is basically what happens. Darwin Core, the standard to save this in the databases, the goal is to facilitate the sharing of information about biological diversity, because it was to control a little bit this heterogeneity of the sources. And it provides permanent identifiers, labels, and definitions so that everyone who works with data on the field can create clean databases. It's maintained by GBIF and the working group. And we have references guides. This is full of links. So you can see sometimes it can go until it's like 300 possibilities. Sometimes you make a download, it has 300 variables because the names include who collected, when did they collect, some notes about, like, there's so many possibilities of information that you can have, that you have a large list of possible variables. And these tables, I know that Sarah told you about keys and how sometimes you have, for example, the species names. Do I have here? Like, for example, you can have a database with a taxonomy and only one, like every name in the list, and it's going to be, it will tell you, for example, the synonyms. And it's one table with the names, but the table with the exicata, with the actual specimens, have these names repeated. So you have to cross the tables because you are not going to be dealing with the whole amount of data. You can go back to the computer. So sometimes you have here the types of and specimens of species are a database in itself, because what was the description, the original description of each species is a special database. For those who are not biologists, every name, every species has one original type that is like, this is the first time we saw this species, this is when we named it, and this is a whole different database. You have the occurrences, you have the, like, every collection of the species, and you can have peripheral databases like the vernacular names, how they are called in every country, which are the references, the literature, the publications where they were referenced, what is their distribution, the regions, the habitats, the vegetation types, this kind of nodes. And at the end you have species profiles. So you have, at the end you will have to deal with different tables, and they come in tables of tables, like groups of tables. The part where we work with relational databases is going to be important when dealing with this data, when cleaning this. We also have some metadata. In addition to Darwin Core, we have an ecological metadata standard. So there are some variables that are noted in this language, in this standard. It's just a way to homogenize all the notation. We will have common columns for each table, for example, the number of the identifier, and this is how you cross the tables. I was talking to Thalias before about the IPT. The IPT is the Integrated Publishing Toolkit. Every collection sends an IPT file that has this structure of several tables that goes up to GBIF. And this is the kind of file that you upload when you upload your own data to. GBIF has workshops about uploading data. Every once in a while they say like, hey, let's upload our data. If you have a field, if you have a paper, they help you organize the data to go up. The important thing is that data sets and species are giving a permanent identifier and citation, so it also helps tracing where does the data come from. So for example, all my examples are from the Rio de Janeiro Botanical Garden, because it's Brazil and it's hugely diverse, not only because I was there. It's like, the Rio de Janeiro Botanical Garden is the largest herbarium in Brazil, which is the most diverse country in the world. And they do have this system, this integrated publishing toolkit. They use their IPT and a version of the whole database of the whole flora of Brazil works, every upload has a DOI, has a link. They have a version name, and this number is growing all the time, and the taxa is also growing. When we say taxa, we're talking about the names, and we, like in general, like genera and families and other taxa, and this is species. And subspecies, for example, subspecies are part of the taxa. And we have this kind of information, and the package that reads IPT data in R is called Finch, but we won't use it today. We are going to be using only download, not direct data. And about data cleaning, before we start using R, I don't know which time it is. It's okay. Just to let you know, especially for those who are not biologists, as I was saying before, names change in time. A species that was described before can be merged into another species. Species can be split in several species. With the advent of molecular biology, some species and some whole groups change families. Some families were merged. So not only the change happens at the species level, it happens sometimes at the whole family level, like the position of the families along the tree is changing, and some groups are still uncertain, so they will keep changing while we have more and more data. But also, think about it, the name of places change in time, toponyms change in time. Your city didn't have the same name 200 years ago. Cities change, the states change, countries change. So every collection that happened in the Guanabara state does not exist anymore, but you have to know where that is. Or where that happens when it was, when it was the Cismarias here, or when it was in the Nueva Granada. When it's say Nueva Granada in a collection, you have to know. You have to find a way to know where that is. So we need gazetteers. There are gazetteers that will help you see the name of the toponym and the current location of this place. Also, there are lots of collections that are like 3 kilometers after the door of the fazenda. I don't know how to say fazenda in English. Like, it's a farm. The large farm of the family cheese. So it's you have to know where that is when it comes from historical recollections. And this is huge work. It's super fun, but it's enormous. And you can have identification errors. I was very bad always to identify, so some of us don't even touch this part, but the people who put the names, they can have problems. And you can also have geo-referencing errors. The person puts a locality, but they forgot to put the point in the GPS. They come back to the hotel, they say, hi, we don't have the point. Let's do this. Chick, this happens. And another thing that happens a lot is that the coordinates that are there are like the closest city or the centroid of the polygon of the state. So if you look at the collections in every country, you have, in Brazil, it's like a point in Goiás is the most diverse point in the country because lots of errors happen. Instead of putting NA, instead of not putting a number, it stayed in the center of the polygon. So when you check the data, you have to check for the centroids of the places. The centroids of all the states tend to have more diversity because there were errors while doing the databases. It happens the same in Colombia, Puerto Lopez, some place around Puerto Lopez, all the points fall there because it's somehow the centroid of the country. And that happens, but when you study, like I would have to be aware of the centroid of Argentina if I study, so you have to have this more automatized. We also have lots of missing coordinates or localities, especially with historical data that sometimes are the most important data. And we have lots of typographic typos, like names that are badly written, and places and names like all the time. It's human error at the end. And when you take a group of people to digitalize, to make a digitalization, to type the information into a database, there's another source of error because people are tired and they're just writing. So you have to double or triple check that. When we check, we can have spatial errors. So this is real data. Sometimes when you map it, it's falling in the sea. And it shouldn't. It's not marine data. We have this a lot. 0 latitude or longitude. It has one of the coordinates, but it does not have the other. And lots of them have 0, 0. Instead of having no data, they put 0, 0. So there's this crossing of the 2 zeros that has lots of points that shouldn't be there. So you have to clean that. And the part of the names, like taxonomic, the original versus the accepted name. For example, here in Brazil, like every country has that. I know the example for Brazil. Brazil is doing a large effort that gathers 500 specialists that are reviewing all the flora in the country. So their names, sometimes they change. They change, but the time that it takes for a change that happens today to go into the system and to be uploaded is sometimes it's months until it goes to GBIF. So when you work locally with data, you can say, I will accept this taxonomical backbone, I will accept the local backbone, and I won't accept what GBIF is telling me. If I have a doubt, I will give priority to this backbone because I know this is happening. But those are conscious decisions you have to make. Yes, and so this is other kind of errors. You can have duplicate data, you can have duplicatas, the fact that you send the same species to different collections. And we have lots of problems with measurement units because meters and degrees and it's not. And with signs like minus the coordinate. We are here in minus, minus. We are in the west and in the south. But some points are in Africa or are like positive, positive. They fall in Europe. This kind of happens. So we have to check for the format, the cleanness, the competition, and we need tools that are automatized. We as a field, we did this by hand. But now we have tools that do this. The good practices, and we always insist in the good practices. The same way that you'd never touch raw data. You only flag wrong data. You never change it. You only flag it, and you make additions, and the corrections are in new columns. And you document all the steps that you made while cleaning, so to ensure reproducibility. And as a good measure, you cite packages and cite the data source. And you have to know the limitations of the data, basically, because there are some things that some species are not going to be easily detected in the field. Sometimes you study a group that it does not fall in the sampling schemes of people. If you have a group that is difficult to detect, you have to know that. If you're working with orchids, orchids are extremely oversampled because they are extremely charismatic. So you don't have an accurate notion of the true diversity of some groups because they are undersampled or they are oversampled. And this is something that you have to start understanding that it happens with diversity. Some plants are so common that people do not sample them anymore. And when you see the maps, it's incomplete maps. Everybody says like, oh yes, it should be everywhere. Every herbarium must have it. And then when you see the map, it's holes because nobody takes it anymore. Everybody has the distribution in their heads, but you don't have the data. But also if you ask a collection to save an orchid or to save a commonly distributed plant, it will take the orchid. So it's also like lots of things involved. I'm going to stop here and we go to the tutorials. We are going to basically make a download, check some errors. I will go with you. I don't know how I will stay here. And we can do it together. I don't know how you did it. And if you have any questions, any comments, any anecdotes about this kind of thing, you can please, while I set up. Is it okay? Claro.
Es que te preguntar si le gustaría más plan de ejercicios de practicar esto más facilita yo no idea de que realmente como cosas para uno no sé, tienen que hacer esto como lo harían
Sí, ok, bien.
pues es que no sé cómo va a ser el examen entonces para poder saber de pronto cómo practicarlo y no cómo solo tener la solución sino intentar saber
Sí, entiendo.
de pronto echar una práctica.
This is not the tutorial. Wait. I am here in the repo that looks like the repo that you have locally. So I will create a whole new script. I will change my. Are we okay? Epa. What's if I say what's your name, it does not say anything. Like what's? Oh, sorry. David? Ah, David. David was telling me something like we are putting the whole tutorial and in the course, and of course it's easy to just run the code. I have done that, but if you go step by step and I will make some, I don't know, some exercise that goes through a whole workflow. But yes, but at least the fact that the tutorial is running, just please don't go through it. It's good that it's running, but I will open it. I have a change here for mine. Where am I? So I hope you installed GBIF, Taxostand, Coordinate Cleaner, and maps. I will just open the tutorial here. And I want you to open the help of the Ox search. I won't do this here. I will do this here. Library. Our OpenSci has other packages that have access to other sources of occurrence. So for example, package SPOCC, it looks for other sources of data, including citizen data repositories. Today we're using RGBIF. And I would like you to check the help, because what I did here was setting a species, and it's Myrsine coriacea. As I told you, I have no, but Ox search is not exactly a very intuitive function, because if you do this, Ox search, Myrsine coriacea, you have to specify that it's the scientific name, and you have to go via the limit of the data, because if you see the help of the package, I don't know if you're seeing this, some of the parameters that they have is the limit. So it has lots of parameters that are GBIF columns, like the taxon key and the publishing country. So you can make search only for the country that published the data, or only for the country of occurrence of the data. And one of the parameters is if the data has coordinates or not. So historical data will go out of this search if you ask it with has coordinate true. If you want only type specimens, or if you are looking for a specific record number, you can ask for this. You can ask only for continents, so there are a lot of parameters about this. And one of the key parameters is the basis of record. The basis of record is one of the parameters that makes possible the distinction of the type of collection that was made. So GBIF holds fossil and human observation data and living specimen data. So if someone is in a zoo, GBIF may have it, but it's not necessarily the kind of data that you want. Or living specimen or machine observation, or so when we talk about collections, we want preserved specimens. So the tutorial says scientific name, but it didn't ask for preserved specimens. So you might want to add this, like basis of record, preserved specimen. And the other thing is the limit. If you do the download, if you do the basic download, if I check here, my friend. The limit for download is 500 records. Why don't you? So you have to open it to the limit. So there's a hard maximum of 100,000, and the default is 500. So if you do not change the limit, it will just take you a portion of the data. So I will just do it with you. Let me know when you have the data downloaded and which is the number of rows that you have. If you have this 568, that's okay. This is the number of. GBIF will close our IP if we do lots of queries, the same query repeatedly, the same species. Sometimes it does, actually. Sometimes it just shuts you out. And remember, you're in your repo, so I'm saving everything in data raw and as a .csv. Okay, so I insist in creating create data raw because all the lessons are in different folders, but you don't need to do this. You're in the same repo that you have, as long as you save in data raw. So if you are with me with the scientific name, everything is good? Can I talk here? Okay. So look at the amount of names that come from a single search from one species. And most of them are names that are no longer in the collections. It's because the names were changed and even the genera changed. Myrcine was Rapania, and especially there were a couple of Rapanias that they were like super large, so there are lots of collections that are called Rapania, and they became Myrcina recently, more or less recently. So we have 37 different names for one name. Go, yes, please.
Okay. I have a question. Why it can have so many changes in the name of a species?
Why? Because it's a question of how taxonomy and systematic works. Sometimes the species are collected in different places and they are called by different names by different people, but it's the same species. So a third person will check all the collections and will conclude that it's the same. So the names are valid, but the person who described the same species first has the priority. So all the specimens that were collected under different names, they go under the new accepted name. So you don't have things that should be the same that have different names. Sometimes the opposite happens. You have a large species that looks like a complex of plants, for example, that look more or less the same. And when some specialist goes, he says, no, they say it's not. This is actually a different species, and this is actually a different species. So one name becomes two. When one name becomes two, sometimes one of the daughter clades keeps the old name, and the other has a new name. So you just like splitting a group of plants from the original name. And sometimes no, sometimes the whole has to change. So it's like 3 or 4 names. So this is part of the story of 200 and 300 years of checking. Somebody at some point, and it's not rectilinear. It's not straightforward. Sometimes it goes via very weird stories of people meeting and people opening collections and finding things. So it depends on the tax. But the priority is for the older person, the first person that gave the correct name. And if you see this column, Myrcine data taxonomic status, you will see that that under the backbone, the current backbone, 2,023 specimens are correctly named. And the others are not correctly named. They should be renamed. They should be under one of the accepted names. You can check this here, for example. This is the same list of names. And the only accepted names are these. Myrcine coriacea with the correct author. People who used Myrcine coriacea with authors that are not correct names because the author is part of the name. And we also have subspecies that are correct names. But the rest of the names are all synonyms. Are you following? Are you going just like, okay, if you have any question. The rest of them were synonymized. And you can see some taxa, for example, Rapania coriacea. No, Rapania ferruginea. This Rapania ferruginea was extremely collected for a long time in Brazil and in South America. So you still Rapania ferruginea in lots of collections. You have 1,000 collections under that name. And then a revision said, no, wait, this is the same. This is Mercina coriacea. So it gets renamed, but Rapania ferruginea is still super famous, and you still find it in collections. Some other names are less famous. Okay. if I have this, I'm okay. Yeah. Of course, we're all tired, but yeah. I haven't had COVID. Come. Come. Oi. Bia.
I'd like to ask you something. Is that I really couldn't get was like the accepted and the synonym, and like something here. Like I understand that they are all referring to the same thing, but how do you like get to like what you call?
That's how many records from the 5,000 records. How many are having these names? So, were you expecting like a 1, like it's accepted 1 or 0 or something like that?
Okay, but how do you know if it's an accepted or a synonym? Like in the data, like how it says like okay that's a synonym.
Yes, yes. They have a taxonomic backbone. Like part of the databases is you have the whole name, like you have a list of the whole amount of names, and the whole amount of names includes names that are correct or not, that are accepted or not currently. So let me show you.
That changes over time, right?
That changes all the time. So you have versions of this. When you have the tables, one of the tables that compose a database is the whole amount of names, and which is accepted or not. But also one of the columns is if you're a synonym, you are a synonym of what? So which is your accepted name? So you will have, for example, Myrsine coriacea and Myrsine, I was talking about Rapania ferruginea. So you have the name. The field is scientific name. And you have other Myrsines. You have Myrsine, another name that is not good. I don't know. Ah, okay, you have Myrsine ferruginea too. And you have lots of records with the same, and you have lots of records with the same, and you have lots of records of the same. You have 37 names, but you have some 5,000 records. So it repeats. And one of the columns is scientific name. You can check in the table, and it says status, taxonomic status, and it will say this is accepted, this is a synonym. But one of the columns is accepted name. And here it will have an A because he's its own accepted name, but here it will say Myrsine coriacea. And here in all the rest of the table, it will say Myrsine coriacea. So when you cross the table with the names that has all the names, wrong or not, with the accepted name key, at the end, if you merge it, it will have only the accepted name. We will do this this is one of the steps. What TPL is doing is that, are you following the part with the taxon stand? I am having trouble with the live coding part. Como es que era? So, when using taxostand, I am not using the whole amount of data because it has 5,000 rows that are repeated. So this is why we're extracting a unique vector from the scientific name. So Myrsini data scientific name has 5,000, but the unique vector will give you only the 37 names. So the species names is the only information that has. So we will have a data frame with 37 original names, not 5,000. This is why we do this. And when we do TPL, of course. What? So you must have had this kind of step. I am only in TPL. I guess you are, some of you are far advanced, but I want to talk about this. TPL will give you some of the errors that it has, some of the comments, and it's mostly about the authors because it does not match exactly, and this is one of the parts of the problem that we have. But other than that, TPL just returns a data frame that has new columns. So tax check. As you can see, TPL did not modify, but it creates new columns that are marked new. They are flagged if the species needed some correction. You can check with view here. So taxon is the column of names that you gave. It was 37 names. And it split internally the genus and the species and the infraspecific rank, the subspecies, for example, and the authors on the authorship. And it went to Tropicus and it went to Q, the herbarium, and check if they were present in their databases. And they returned taxonomic status, if accepted or not, and their family or not. And when it was necessary, they gave back a new genus. When it did not, for example, you have here, Rapania lancifolia. Rapania lancifolia is considered accepted, but it's still considered Rapania lancifolia. And we have to check if this is true or if for some reason the taxonomy is changing. So if we believe in the Brazilian data or not, for example. But what it did is creating new columns, and they are called new. So new infraspecific rank, new species, and new authority, the new authorship. Okay, so we have a table of 37 rows only, and we need to put them back into the data, and this is what merge is. And this is something that you did already in past exercises. Let me close this. And what we create here is a new data frame with the new taxonomy. And what we are doing here is that we are creating the species names that we gave to TPL. We are calling it scientific name. What this will make is that we have a column that is common between the two data sets. So we can merge by the new scientific name column, okay? And then we save the data. I'm just going to execute it here. New text. One of the ways of doing this also, I know you didn't see a lot of dplyr these days, but merge has the full joins and the left joins. And you saw the left joins and the full joins. And how we would do this in dplyr would be doing, if we have a longer table, we have a table that is long, and we have a table that is short. It's the 37. If I do a left join, it will take every line here, and will add the data from the 7. So this would be a left join from the large to the short. So this should work. From Ircine. And new text. And what it does is that it already tells you by what variable they are joining. Sometimes it's more than one. Sometimes they have lots of columns in common. And the dimensions should be the same. And you can see it has 191 columns. This is the heterogeneity of the data. And I was telling you that we were going to do some maps. I guess that this command worked for all of you. Did you have the map? Okay, perfect. So map is a function that comes from package map. And this is part of the old school maps in R. It existed for a long time. And it's weird because it has 3 virgulas. No, 3 commas, sorry. I do that all the time. Map. Did you check why do you have to write map like this? So what we're doing here is plotting the decimal latitude in function of the decimal longitude of the data that we have. And I specified a small parameter that is the aspect. You can try to do a plot without it. If you took out aspect, it will decide which is the correct. It will decide that this is the way it plots. Given the space available, it won't care if here we have a 10 interval, a 10-degree interval, and here we have a 51. It is not respecting an aspect. So we don't have lots of, so we will have like not a way to map. So this is why I put aspect equal true, equal 1. So it will rescale accordingly. Yes. And the map, I was telling you that map is one of the old school ways to map. I like it because it's super quick. You add the map on top of anything. But basically, you use the three commas to call it, because the default is already the database of the world. The second is it doesn't care about the regions. It doesn't care to be exact. So you can just omit these first arguments and just tell it add, please add equal true. Add equal true will allow you, it's false right now. The default is not to add anything. So you have to change this default. Yes. But I wanted us to make an SF object, like I told you yesterday. We don't have a lot of time, but I would like to ask you something that is not in the tutorial.
Hi.
Sure.
The second code to the second map, this one, the scale is not the same.
That here. Because I didn't put. Yes, that's true.
This is not the output that
you have.
Yeah, the second one, it's another scale.
The second one is different for you.
Yes.
I guess it's because the cleaning. I am having, for some reason, that the cleaning is only from the region in the Andes. Let me see what you have. The scale, yes. Okay, that sounds better, but.
Yeah, the scale is not.
When we change the scale, this is changing here too.
No, it's because the data set is different.
Yeah.
It's what is considered cleaned in the cleaning that you did, and the cleaning that I did is different.
Yeah, and the third one is different as well. The rest one as well.
Okay, one of the things that we should be doing, like we have some extra columns in the new data frames, and they are accordingly to the steps of cleaning that coordinate cleaner did.
Besides this, when we change the scale, look, you can see the US, right?
Yes. Yeah, it's because it's
not fixed.
No, it's not fixed. It's using the device that you're giving it.
These points are not the same as well with the other that you sent.
because some of them are cleaned. Did you make the download for the only preserved specimens, or only the? I think maybe this tutorial is all with the preserved specimens. I would have to plot the Mersini cord data set. I don't know why I have this cleaning extra. Do you have the dimensions of this second data set? What? Do you have the dimensions of this second data set, the number of rows? You can. No, but you can ask it, give me the n rows or the dimensions of the second data set, of the clean one, yeah. No, yes, the second one. No, but the dimensions of the data frame, the number of rows and the number of columns. Geo-clean, geo.clean. No, the second one is the second. Yes, this is the original one. Geo-clean, yeah. 2, 4, 6, 6. So I am having a different. No, look, no, it's good. It flagged 104 of 2000. It didn't flag a lot of. There may be something. Yeah. Yeah. And of course, it's when you do this, it just does not like to map. Maps, map.
It's the same here.
Yeah. Something happened during the knitting? I have no idea. Yeah. So what I wanted to do, you have a data frame, and you have been only plotting the data frame. And yesterday I talked a lot about SF objects, and the tutorial is finishing at a part where we are writing .cbs to the disk. So the next step, and it is something that you would like find at home, is the following thing. How do you create a map similar to this in Tmap? So for this, you have to find a way to. I told you yesterday how to do a quick map. It was basically data world, and you added the TM shape, and it already had the basics. Okay? How do you add a shape with these points and add some points to that map? It has a different structure, it has a different syntax. What you will need to do is take this data frame currently, that is and convert it into a shape file, into an SF object. So if you remember, and this is just to finish, if you remember, I told you yesterday that SF objects were very similar to data frames. The only difference, one of the key differences, is that SF objects have a column that is called the geometry. Yes? So the geometry in SF is somewhere here. It's in decimal longitude and in decimal latitude. Yes? So how do you go from a data frame that has decimal latitude and longitude to an SF object where geometry is these two columns? So this is step one. You create an SF object from this. And step two is having something like Myrcene SF that is an SF object. How do you add it to yesterday's map? How do you add it to yesterday's map? The map that we made in Tmap, that was very quick, that was like South America. You filter the world, continent, South America, ploop. How do you, the next step of this, it's the same code will be the beginning. You added a shape, and now you will have a shape. So what are the next lines to add the dots to a Tmap map? So this is something that you resolve when you, and make sure when you do this at the end to explore something that is called the Tmap Mode. The Tmap Mode allows you to create only plots or interactive maps. So when you finish your map in Tmap, execute Tmap Mode and try to have an interactive map of these points and explore it. Okay? So this is for you to stay with it. Yes? So because you have already the data frame, a clean data frame that is flagged. So make sure that you explore what happened, which are the steps, the new columns, but especially try to create a map similar to this one that was super quick in the background, in the framework that I showed you yesterday. It should be not very But it will require you to go through the help and maybe to remove some NAs and this part. And we can talk about this tomorrow. Yes? Okay. Thanks a lot. Do you have any questions? Do you have any comments? Do you have any? Cool.
To do the add commit, should we include the data? Because like here we have the instructions to download the data.
I would include the data. I would include the data. It's CSVs. It's CSVs. Yes.
Okay.
Yes. It should be. It should be. I don't know what happens. Like what happens with me with the scripts is that I always code very messy, and when I do the commit, I clean it a little bit. But when you do the commit, you put the script and add the script for this SF of the Tmap when you get it. It can be a second commit, and add the data. So something about the commits every day is that we talk a lot about the commit of the day as if it were one commit of the day. So of course it's good to have one commit per day, but it's absolutely fine if it's 15 commits, because sometimes you go back and forth, you correct. So the story of the commits does not need to be clean. You just go forward and you correct, and sometimes I forgot this, so you commit extras, that's okay. So the commit of the day is the commits of the day. But yes, add the data, add the two data sets that were created, the one in process, the one in raw, the script of the tutorial, and after that, the Tmap. I want to see Tmaps. I want to see interactive. And you can explore like colors and things, and of course. And tomorrow we'll talk a little bit about diversity. Yes. So it's over for me, but you can stay if you want. Thanks everyone.